Good afternoon, everyone. I'm Jishnu. I'm an assistant professor at the University of Pittsburgh. I run a computational systems immunology lab. We use machine learning and network approaches to study immunomodulation in a wide range of contexts, from infectious to autoimmune diseases. And we do this really in a principled way, trying to look at both protein-protein interaction networks and gene regulatory networks. And what I'll talk to you about today is our work with gene regulatory networks in the B cell system. So this is part of a large consortium. So we are one of the centers in a National Human Genome Research Institute funded consortium called the IGVF consortium, also the impact of genetic variation on function. And all the work that I'll show today is done with my close colleague and collaborator, Harinder Singh, who he and I have this grant, and this was co-led by people in his lab, Nick Pease, Jingyu Fan, postdocs in his lab, as well as Zarife, postdoc in my lab, Swapnil and Akansha, graduate students in my lab. So this really represents a large body of work done through this joint grant that we have. I won't have time to go through all of the details, but I'll focus on the key pieces that are hopefully of interest to this RegSys audience. Specifically, how using this B cell system, we characterize state-specific gene regulatory networks. So now an introduction to the system. So this is all an in vitro system which tries to match what happens in vivo. So naive B cells, upon antigen encounter, undergo activation, and they undergo a primary bifurcation into germinal center, like B cells and extrafollicular plasmablasts. Then they undergo a secondary bifurcation into memory B cells and plasma cells, specifically long-lived plasma cells. All of this is captured in an in vitro system that mirrors antigen encounter in an in vivo setting. And what we have done is we have tried using multi-ohm data to construct state-specific GRNs, coupling it to interpretable machine learning, figure out some novel biology underlying this bifurcation event. So like the first part, and this is really two manuscripts, and this is hot off the press. The first is under review, the second is unpublished. There are a lot of tools for GRN inference, but the goal of this talk is hopefully to tell you that we have this new way of thinking of cellular programs that, coupled to traditional GRN inference, give you novel insights. So this is simply a review from Julio Says Rodriguez that says that there are more than 20 plus methods for GRN inference, and we also did our own GRN inference, but with a twist. So what we did is we did a two-step GRN inference. So CellOracle is a method that many of you may be familiar with that uncovers GRNs. What CellOracle does is, first it comes up with a base GRN, very simple ideas that most of this community will be familiar with. They look at things that are co-accessible with the promoter, then they do motif enrichment. That's the easy part, the base GRN part. Then they fit context-specific GRNs using a Bayesian regression model, Bayesian ridge regression model that gives you now TF gene links for each specific cluster. What we did is we slightly changed it. We kept the context-specific GRN the same. We said, okay, we are fitting the expression of a gene to the specific TFs that could possibly regulate that gene in that cell state. That's the second part. But the base GRN doing TF motif enrichment is really basic. It doesn't capture actual binding. So we changed it around and we said that we will get CRE gene linkages from this probabilistic topic model in MIRA, and then using actual binding data. So we have state specific. So I talked to you about 5 states: naive, activated, germinal center, plasmablast, and memory B cell. So we have multi-ohm data. So we have all of this is cell state specific. The only thing that is non-specific is the binding data, which is from GM12878, which is a tier 1 encode cell line, which is mirroring an activated B cell. So we did this two-phase construction of GRNs, and as we described in this marker paper from the consortium, we adopted a map, predict, perturb framework. So we have now mapped these GRNs. What can we predict? And when we actually perturb, do these predictions hold up? And this is the preprint for the first manuscript in case you're interested. So we use CellOracle to do in silico perturbations, and we started off with a bunch of transcription factors that are lineage defining. So for example, PAX5 is known to be lineage defining for the germinal center fate. If you knock out PAX5 in the primary bifurcation event from an activated B cell, you would now not get GC-like cells, you will primarily get extrafollicular plasmablasts. That's exactly what we predict. There's an outflow from the GC into the PB. If you do play this game on the other side, PRDM1 or BLIMP1, if you knock this out, it moves out of the PB fate into the GC fate. All of this is good, but how accurate is this in an ensemble setting? And to do this, there were TF perturb-seq experiments done. So perturb-seq for 5 transcription factors, but phenotypic readouts for 30 odd transcription factors, each of which bifurcates into a GC or a PB-like fate. And we compared actual experimental results with the in silico perturbations, and we believe this is the first instance where this has been done this systematically. We see a very nice correlation of around 0.7 between the experimentally measured phenotypic GCPB bifurcation and the Cellular predicted perturbation score. What is more interesting is, and this is really assessing the quality of the GRN, is if you now do this with the standard way to construct GRNs using Cellular, this correlation drops from around 0.7 to 0.4, meaning that because Cellular is using network propagation to actually propagate the influence of these perturbations on the GRN, it is directly based on the quality of the network. And we clearly see that by using this better GRN, where we replace the base GRN with MIRA, add in binding data from SysTromDB, we are getting better linkages. So now we have a good network. There's a lot more in the first manuscript, which I don't have time to go into, but I'll now get to the interpretable machine learning part. And how can we couple interpretable machine learning to these state-specific GRNs to learn something new, hopefully. And to do this, we applied a method that we published last year in Nature Methods called SLIDE. It's a latent factor regression approach. I will go over this very quickly. So most methods link observable features, the X's that you measure, and this is a very generalized idea. The X's could be anything. It could be transcriptomic, epigenomic, proteomic, metabolomic. Obviously, in this case, we are measuring transcript abundances, but this is a general framework that basically says that most methods link X's, observed features, to responses of interest. However, biology doesn't care about what we can or cannot measure. There are these latent states or hidden states, and we want to first go from the observed features to the latent states and then regress using the latent factors to find a set of necessary and sufficient latent factors. The idea of latent factors is not new. Latent factors have been around in the statistical literature for a long time, but what we came up with was unique in 3 different ways. First, most latent factor methods do not have identifiability guarantees, meaning they are not uniquely identifiable. So things like non-negative matrix factorization, you have an infinite number of decompositions. On the other hand, if you use things like principal components regression, you make orthogonality assumptions. Here, we were able to prove unique identifiability guarantees without making assumptions about orthogonality. We solve this as a basically, we have a constrained optimization problem where each latent factor is anchored by 2 features, at least 2 features. Everything else can be shared or not shared in a data-dependent fashion. Because we now have latent factors that are overlapping and yet stable, we can figure out what is necessary and sufficient using a method for overall false discovery rate control. Adaptation of knockoffs. Knockoffs is something developed by Emmanuel Candace and colleagues. The obvious question is, why are we not using a standard regression setup to do this? Because of multicollinearity. The Z's, the latent factors, are multicollinear. So you would have deflation of p-value. So here we use this method for overall false discovery rate control and say that this is the necessary and sufficient set of latent factors. That's innovation 2. Innovation 3 is we use a nonlinear model. So we are not just using linear terms. We first identify the significant standalone factors, then we identify significant interacting ones. And the importance of this will become clear as we go through the GRN part. And we've used this in a wide range of contexts, which I will not go over outside gene regulatory network inference. But here the question really is, can we now couple SLIDE to the analyses of these state-specific GRNs to gain additional insights into regulatory mechanisms? So the first thing that we did is we looked at these two bifurcated states, the germinal center state and the extrafollicular plasmablast fate. And we came up with two latent factors, one significant standalone factor and one interacting latent factor that can discriminate these states. In terms of discrimination, just the linear model, the standalone factor, is enough. But the interaction gives us something in terms of inference, which we will get to. So now when we look at these two latent factors, and the talk is entirely hinged on coupling these two latent factors to the state-specific GRNs that we inferred. The inferred latent factors are stable and meaningfully capture bifurcation trajectories. You can learn this from the single-cell RNA part of multiome and go to RNA only, or do it the other way. It's all stable, which is good. Often doesn't happen with the approaches. It also captures pseudotime trajectory. But this is all standard. So what new biology did we actually learn? So the first thing that we said is, can we now couple them? One is we have the standard path, but with some innovations, we have these high-resolution state-specific gene regulatory networks. The other is we learn these cellular programs with a lot of statistical guarantees. Can we now see the slide part only uses RNA. It has no prior knowledge. It does not use the attack data. It has no prior knowledge of any TF motifs or anything along those lines. However, do we see specific regulons enriched in the slide latent factors? And we actually see about 15 TF regulons enriched in these cellular programs. A lot of these are well-known lineage-defining transcription factors for these two fates. So we are capturing new biology. But the more interesting part is, I talked about this interaction term in the nonlinear model, which mathematically is simply an element-wise product of the two matrices, but it is now capturing something about GRN architecture that is non-trivial. And remember, we had two factors. So the latent factors are identified based on the covariant structure. So correlation of expression and co-regulation should be synonymous. That would be within a single factor. But the interacting terms are two completely different latent factors, and we are picking up regulons across these two factors. So the interactions of the nonlinear relationships are picking up some non-trivial regulatory relationships. So we then looked at things like scenic plus, and what we see is we get far more specific regulons than scenic plus. Scenic plus regulons are great, widely used by the community. We also use them. However, they are large. And for actual experimental testing, when we look at things downstream of these transcription factors, we just look at absolute value of fold change for the genes across the two states. What we are picking up is much higher effect sizes, both overall and in terms of specific transcription factors. And this is something that we can see across complex differentiation systems. So I will not belabor this point, because this we've done also for T cells in a chronic versus acute infection setting. I will not go over the details in the interest of time, but this is just to say that this is not a one-trick pony. It can actually work for complex systems. But today's talk is focused on B cells, so I will stick to that. So the next thing that we said is we have static state-specific GRNs. What if we move to dynamic GRNs? And here we used a method called Dictis, developed by Luca Pinello's lab. Luca is here. This is really a fantastic method. So dynamic GRN infers regulatory circuits over continuous time by extrapolating across time points. So we have measured this at specific time points. But now if we extrapolate, can we get a dynamic GRN? And what do we get in terms of the dynamic GRN? So we had things that we prioritized, enriched TF regulons that we prioritized using the static GRN. Now, when we couple it to the dynamic GRN, what do we see? And we have this for the two primary branches that I talked about, the germinal center branch and the plasmablast branch. In the PB branch, we see that the lineage-defining transcription factors, the ones that we picked up, have changes both in terms of expression dynamics of the transcription factors as well as regulation dynamics over waves. So I won't go through the underlying biology, but we recapture a lot of things that are known, like IRF4 and PRDM1 talking to each other, PRDM1, XPP1 becoming very, very important in the late stages of PB development. But we also find some new things, which I'll come to. We see the same thing for the GC branch. We capture both expression and regulation dynamics. But to really understand this, we have three different modes. We have a static GRN, we have a static GRN now extrapolated dynamically. But what if we did not have the static GRN? What if we only looked at a dynamic GRN? What would we learn? And here, this is basically saying you have the activated state and you have the final bifurcated PBGC states. But you now have episodes. So you can pick up episodic enrichment of TFs even within this. And when we do this, we find a lot of transcription factors enriched that are actually missed when you look at the endpoints. You only get this when you look in the middle. And we've actually looked at this systematically. So different modes of TF activity, when you look at what is enriched in the static state and in the dynamic episodically enriched, the overlap is quite low. So we are picking up discrete things. But I'll focus on one novel discovery. I said we found a lot of things that were known, but one novel discovery was this transcription factor called CREB3L2, which showed up as enriched in the static. We see differences in expression and regulation dynamics, and it also comes up in episodic enrichment. And this was not at all known to have any role in the fate bifurcation GC versus PB. This is a system that immunologists have studied for over 2 decades. And CREB3L2 knockout does not impair the generation of plasmablasts, but it does impair PC survival over time, showing that it really has a dynamic fate. So people did not find this because when you look at the early extrafollicular PBs, it does not have an impact. But over time, and what I'm not showing here is antibody secretion over time is impacted. So it clearly in the dynamic development bifurcation of these fates. The final point that I would end with is, can we now do something with just slide, this interpretable machine learning approach that we developed, or do we need GRN? So we said, well, what if you take away the GRN? So we thought experiment of rolling back cellular programs in time. So now you have lineage defining transcription factors. So you have, let's say, IRF4 and PRDM1, which basically are important for the PB lineage. So when you do a TF perturb-seq experiment, when you knock them out, you should have more GC cells and vice versa. So we now said, can we learn, and here there is no GRN, can we learn a cellular program from the TF perturb-seq experiment, the knocked out versus the untreated cell? So when you knock out PRDM1 or IRF4, you have a GC-like cell. If you compare it to true GC cells, how good? So you knock this out on activated B cells about to undergo the perturbation. At day 4, you are measuring the expression readout, and then they are bifurcating. But when you artificially knock it out, it now goes mostly towards one fate or another. If you learn a cellular program from this, can you now roll back to day 2 and make predictions for things that are pro-GC and pro-PB? And when we do this with standard methods like DEGs or lasso, you basically make an all or nothing prediction, which is essentially useless because you can't discriminate. But when you now do this using slide, both for PRDM1 and for IRF4, on the top you see these yellow knockout versus untreated cells. You learn a cellular program and you predict that about 25% of cells, these are unperturbed cells that are GC-like, and you get a similar number but a different model for the 2 different transcription factors. And when you actually compare them to true GC, they are almost indistinguishable, exactly as it should be. And when you compare them to PB, it's night and day. So they are transcriptionally similar to true GC cells. And the cells that you get from these 2 transcription factors acting together are much more phenotypically similar than the irrelevant TF perturbation knockouts. So you're basically able, using interpretable machine learning, to really roll back cellular programs. And this is just one system, but we have done this for other systems too. So in summary, we reconstructed high-resolution state-specific B-cell GRNs and used a map predict perturb framework. We couple these to slide to identify key regulons that would be missed by conventional network topological analyses, the inferred cellular programs, stable and capture bifurcation. We also identified a dynamic GRN and coupled slide to both the static and the dynamic GRN to unveil novel molecular circuits. And finally, we allow the rollback of cellular programs in time to this is really generalizable because now you can predict which cells will undergo a fate bifurcation. And this is the lab, so I get to talk about all the wonderful work that they do, but it's really all of them doing the work. So very fortunate to have such a great group of people, and I would be remiss if I didn't thank my funding sources, the organizers for allowing me to present, and all of you for your attention.
Questions? Everyone wants to go on a break.
Yeah, thanks for the nice talk. So I have 2 questions. So first one is when you talk about this bifurcation and trajectory for sales, could you further elaborate on whether this transition is reversible or not, or how can this be biologically interpreted?
Yeah, so that depends on the details of the system. Depends on the details of the system. What I will clarify here is that in this system, we are simply making a prediction of a stochastic initial fate commitment. We are not saying it has made an absolute commitment, but whether it's a stochastic initial fate commitment or a final, that depends on the details of the system.
Okay, another follow-up question is like, here you are mainly focused on the transcriptional activation by transcript factors. However, we know there's another universe of the non-coding RNAs which are also hard to ignore, right? So how could this impact or the work that you're doing, or whether there's ongoing work on that direction? Yeah.
It's a great question. We haven't done anything concretely in that space, but conceptually we can use interpretable machine learning approaches like SLIDE on these non-coding RNA abundances. We haven't done it, but conceptually it can be done. But that's a good point.
No more questions. So thank you, everyone. Thank you again.